Hello everyone, I'm Tanu Sharma from BSc Chemistry Honors. Today I'm here to record a video for visually impaired students on topic drug. Drugs are chemicals of low molecular masses. These interact with the macromolecular targets and produce a biological response. When the biological response is therapeutic and useful, these chemicals are called medicines and are used in diagnosis, prevention, and treatment of disease. Most of the drugs used as medicines are potential poisons if taken in doses higher than those recommended. Use of chemicals for therapeutic effect is called chemotherapy. Drugs can be classified mainly on criteria outlined as follows on the basis of pharmacological effect. This classification is based on the pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for the treatment of a particular type of problem. For example, analgesics have pain-killing effect, antiseptics kill or arrest the growth of microorganisms. On the basis of drug action, it is based on the action of a drug on a particular biochemical process. For example, all antihistamines inhibit the action of the compound histamine, which causes inflammation in the body. There are various ways in which action of histamines can be blocked. You will learn about this in section 16.3.2. On the basis of chemical structure, it is based on the chemical structure of the drug. Drug classified in this way share common structural features and often have similar pharmacological activity. For example, sulfonamides have common structural feature given below. On the basis of molecular targets, drugs usually interact with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. These are called target molecules or drug targets. Drugs possessing some common structural features may have the same mechanism of action on targets. The classification based on molecular targets is the most useful classification for medical chemists. Macromolecules of the biological origin perform various functions in the body. For example, proteins which perform the role of biological catalyst in the body are called enzymes. Those which are critical to communication system in the body are called receptors. Carrier proteins carry polar molecular across the cell membrane. Nucleic acids have coded genetic information for the cell. Lipids and carbohydrates are structural parts of the cell membrane. We shall explain the drug target interaction with the example of enzymes and receptors. Catalytic action of enzymes. For understanding the interaction between a drug and an enzyme, it is important to know how do enzymes catalyze the reaction. In their catalytic activity, enzymes perform two major functions. The first function of an enzyme is to hold the substrate for a chemical reaction. Active sites of enzymes hold the substrate molecule in a suitable position so that it can be attacked by the reagent effectively. Substrates bind to the active site of the enzyme through a variety of interactions such as ionic bonding, hydrogen bonding, van der Waal interactions, or dipole-dipole interaction. The second function of an enzyme is to provide functional groups that will attack the substrates and carry out chemical reaction. Drug enzyme interaction. Drug inhibit any of the above mentioned activity of enzymes. These can block the binding sites of the enzyme and preventing the binding of substrate, or can inhibit the catalytic activity of the enzyme. Such drugs are called enzyme inhibitors. Drugs inhibit the attachment of substrate to an active site of enzymes in two different ways. Drugs compete with the natural substrate for their attachment of the active site of enzyme. Such drugs are called competitive inhibitors. Some drugs do not bind to the enzyme active site. These bind to a different site of enzyme which is called allosteric site. This binding of inhibitor or allosteric site changes the shape of the active site in such a way that substrate cannot recognize it. If the bond formed between an enzyme and an inhibitor is strong covalent bond and cannot be broken easily, then the enzyme is blocked permanently. The body then degrades the enzyme inhibitor complex and synthesizes the new enzyme. Receptors are proteins that are critical to body's communication processes. Majority of these are embedded in cell membranes. Receptor proteins are embedded in the cell membrane in such a way that their small part possessing active site projects out of the surface of the membrane and opens on the outside region of the cell membrane. In the body, message between two neurons and that between neurons to muscles is communicated through certain chemicals. These chemicals known as chemical messengers and received at binding sites of receptor proteins. To accommodate a messenger, Shape of the receptor site changes. This brings about the transfer of message into the shell. This chemical messenger gives message to the shell without entering the shell. There are large number of different receptors in the body that interact with different chemical messengers. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure, and amino acid composition. Drugs that bind to receptor site and inhibit its natural function are called antagonists. These are useful when blocking of a message is required. There are other type of drugs that mimic the natural messenger by switching on the receptor. These are called agonists. These are useful when there are lack of natural chemical messenger. Antacids. Overproduction of acid in stomach causes irritation and pain. In several cases, ulcers are developed in stomach. Until 1970, only treatment for acidity was administration of antacids such as sodium hydrogen carbonate or a mixture of aluminium and magnesium hydroxide. However, excessive hydrogen carbonate can make the stomach alkaline and trigger the production of even more acid. Metal hydroxides are better alternatives because of being insoluble. These do not increase the pH above neutrality. These treatments control only symptoms and not the cause. Therefore, with these metal salts, the patients cannot be treated easily. In advanced stages, ulcers become life threatening, and its only treatment is removal of the affected part of the stomach. A major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical histamine stimulates the secretion of pepsin and hydrochloric acid in the stomach. The drug cimetidine was designed to prevent the interaction of the histamine with the receptor present in the stomach wall. This resulted in release of lesser amount of acid. The importance of drug was so much that it remained the largest selling drug in the world until another drug, Genentech, was discovered. Histamine is a potent vasodilator. It has various functions. It contracts the smooth muscles in the bronchi and gut and relaxes other muscles, such as those in the walls of fine blood vessels. Histamine is also responsible for the nasal congestion associated with common cold and allergic response to pollen. Synthetic drugs, bromopeniramine and terfenadine act as antihistamines. They interface with the natural action of the histamine by competing with histamine for binding sites of receptor where histamine exerts its effects. Now the question that arises is why do above mentioned antihistamines not affect the secretion of acid in stomach? The reason is that anti-allergic and antacid drugs work on different receptors. First, tranquilizers and analgesics are neurologically active drugs. These affect the message transfer mechanism from nerve to receptor. Tranquilizers are a class of chemical compound used for the treatment of stress and mild or even severe mental disease. These relieve anxiety, stress, irritability, or excitement by including a sense of well being. They form an essential component of sleeping pills. There are various types of tranquilizers. They function by different mechanisms. For example, noradrenaline is one of the neurotransmitters that plays a role in mood changes. If the level of noradrenaline is low for some reason, then the signal sending activity becomes low and the person suffers from depression. In such situations, antidepressant drugs are required. These drugs inhibit the enzymes which catalyze the degradation of noradrenaline. If the enzyme is inhibited, this important neurotransmitter is slowly metabolized and can activate its receptor for long periods of time, thus counteracting the effect of depression. Ironized and phenylgine are two such drugs. Some tranquilizers, namely chlordiazepam oxide and maprovimate are relatively mild tranquilizers suitable for relieving tension. Equinil is used in controlling depression and hypertension. Derivatives of barbituric acid with vernal amytal, nambutal, luminal, and second constitute an important class of tranquilizers. These derivatives are called barbiturates. Barbiturates are hypnotic sleep producing agents. Some other substances used as tranquilizers are Valium and serotonin. Analgesics reduce or abolish pain without causing impairment of consciousness, mental confusion, incoordination or paralysis or some other disturbances of nervous system. These are classified as follows: non-narcotic, non-addictive analgesics, narcotic drugs, non-narcotic, non-addictive. Aspirin and paracetamol belong to class of non-narcotic analgesics. Aspirin is the most familiar example. Aspirin inhibits the synthesis of chemicals known as prostaglandins, which stimulate inflammation in the tissue and cause pain. These drugs are effective in relieving skeletal pain such as due to arthritis. These drugs have many other effects such as reducing fever and preventing platelet coagulation. Because of its anti-blood clotting action, aspirin finds use in prevention of heart attacks. Narcotic analgesics. Morphine and many of its homologues, when administered in medical doses, relieve pain and produce sleep. In poisonous doses, these produce stupor, coma, convulsions, and ultimately death. Morphine narcotics are sometimes referred to as opiates since they are obtained from the opium poppy. These analgesics are chiefly used for the relief of postoperative pain, cardiac pain, and pain of terminal cancer in childbirth. Antimicrobials. Disease in human beings and animals may be caused by a variety of microorganisms such as bacteria, virus, fungi, and other pathogens. An antimicrobial tends to destroy, prevent development, or inhibit the pathogenic action of microbes such as bacteria, fungi, virus, or other parasites selectively. Antibiotics, antiseptics, and disinfectants are antimicrobial drugs. Antibiotics. Antibiotics are used as drugs to treat infections because of their low toxicity for humans and animals. Initially, antibiotics were classified as chemical substances produced by microorganisms, bacteria, fungi, and molds that inhibit the growth or even destroy microorganisms. The development of synthetic methods has helped in synthesizing some of the compounds that were originally discovered as products of microorganisms. Also, some purely synthetic compounds have antibacterial activity and therefore definition of antibiotic has been modified. An antibiotic now refers to a substance produced wholly or partly by chemical synthesis which in low concentration inhibits the growth or destroys microorganisms by inverting in their metabolic processes. The search for chemicals that would adversely affect the invading invading bacteria but not the host began in 19th century. A German bacteriologist convinced this idea. He investigated arsenic based structures in order to produce less toxic substances for treatment of syphilis. He developed the medicine arsefonamide known as salvarsan. He got Nobel Prize for Medicine in 1908 for this discovery. It was first effective treatment discovered for syphilis. Although salvarsan is toxic to human beings, its effect on the bacteria spirochete, which causes syphilis, is much greater than on human beings. At the same time, Ehrlich was working on azides also. He noted that there is similarity in the structure of salvarsan and azides. Azides. The double bond AS linkage present in the Earth's phenamine resembles the N double bond N linkage present in azides in the sense that arsenic atom is present in place of nitrogen. He also noticed tissues getting colored by dyes selectively. Therefore, he began to search for compounds which resemble in structure to azides and selectively bind to bacteria. In 1932, he succeeded in preparing the first effective antibacterial against Prontocil, which resembles in structure to compound salvarsan. Soon it was discovered that in body Prontocil is converted to a compound called sulfanilamide, which is a real active compound. Thus, the sulfur drugs were discovered. A large range of sulfonamide analogs was synthesized, one of the most effective in sulfapyridine. Despite the success of sulfonamides, the real revolution in antibacterial therapy began with the discovery of Alexander Fleming in 1929. The antibacterial properties of Penicillium fungus, isolation and purification of active compounds to accumulate sufficient material for clinical trials took 13 years. Antibiotics have either chedal effect or static effect on microbes. A few examples of the two types of antibiotics are as follows: bactericidal penicillin, aminoglycosides, ofloxacin, bacteriostatic erythromycin, tetracycline, chloram, chloramphenicol. The range of bacteria or other microorganisms that are affected by a certain antibiotic is expressed as its spectrum of action. Antibiotics which kill or inhibit a wide range of gram-positive and gram-negative bacteria are said to be broad spectrum antibiotics. Those effectively mainly against gram-positive or gram-negative bacteria are narrow spectrum antibiotics. It effective against a single organism or disease. They are referred to as limited spectrum antibiotics. Penicillin G has a narrow spectrum. Ampicillin and amoxicillin are synthetic modification of penicillins. These have broad spectrum. It is absolutely essential to test the patients for sensitivity, allergy to penicillin before it is administered. In India, penicillin is manufactured at Hindustan Antibiotics in Pimpri and in private sector industry. Chloramphenicol, isolated in 1947, is a broad spectrum antibiotic. It is rapidly absorbed from the gastrointestinal tract and hence can be given orally in case of typhoid, dysentery, acute fever, certain form of urinary infections, and pneumonia. Vancomycin and ofloxacin are the other important broad spectrum antibiotics. The antibiotic dicydegerine is supposed to be toxic towards certain strains of cancer cells. Antiseptic and disinfectant. Antiseptic and disinfectants are also the chemicals which either kill or prevent the growth of microorganisms. Antiseptic are applied to the living tissue such as wounds, cuts, ulcers, and the skin surfaces. Example: the furosin, etc. These are not ingested like antibiotics. Commonly used antiseptic Dettol is a mixture of chloroquine and terpineol. Bithionol, the compound is also called bithionol, is added to soaps to impart antiseptic properties. Iodine is a powerful antiseptic. Its 2-3% solution in alcohol water mixture is known as tincture of iodine. It is applied on wounds. Iodoform is also used as an antiseptic for wounds. Boric acid in dilute aqueous solution is weak antiseptic for eyes. Disinfectants are applied to inanimate objects such as floors, drainage system, instruments, etc. Same substances can act as an antiseptic as well as a disinfectant by varying the concentration. For example, 0.2% solution of phenol is an antiseptic while its 1% solution is disinfectant. Chlorine is in the concentration of 0.2 to 0.4 ppm in aqueous solution, and sulfur dioxide is very low concentration are disinfectants. Antibiotic revolution has provided long and healthy life to people. The life expectancy has almost doubled. This increased population has caused many social problems in terms of food resources, environmental issues, employment, etc. To control these problems, population is required to be controlled. This has led to the concept of family planning. Anti-fertility drugs are of use in this direction. Birth control pills essentially contain a mixture of synthetic estrogen and progesterone derivatives. Both of these compounds are hormones. It is known that progesterone suppresses ovulation. Synthetic progesterone derivatives are more potent than progesterone. Norethindrone is an example of synthetic progesterone derivative most widely used as anti-fertility drug. The estrogen derivative which is used in combination with progesterone derivative is ethyl estradiol novelstrol.